So this lecture will focus on gene regulation. So we'll actually be skipping most of chapter 11 and focusing specifically on modules 11.1 to 11.7. So here are the major points from this lecture and the last. So last lecture we focused on how genes are transcribed to mRNA and translated to protein. In transcription, that was done through RNA polymerase. In translation, that was done through tRNAs in the ribosome. Today we're going to see exactly how transcription and translation are controlled. So this is the process of gene regulation. So regulating what genes are transcribed and what genes are translated. So here's question 1 for Blackboard. So true or false, the sequence of DNA found in cells that make up your skin is the same as in cells that make up your intestines. So here the answer is actually true. So this is actually a very common misconception in biology. So actually the DNA in most of your cells is exactly the same. The DNA in your skin cells is the same as the DNA in your intestines. The DNA in your intestines is the same as the DNA in your eyes. So really there are very few exceptions to this. Of course, the most obvious one would be sperm and egg cells. So those have half the number of chromosomes, as you remember from the mitosis-meiosis lectures, as a normal cell. So for somatic cells or non-sex cells, those have exactly the same DNA as the rest of the somatic cells in your body. So this then leaves the question then of how is a skin cell different from an intestinal cell then? So what makes them different? So this is done through the process of gene regulation. So for something like a skin cell, as we talked about, that has the same DNA as an intestinal cell. So the major difference between the two, though, is what genes are on and which genes are off. So again, how the genes themselves are regulated. So as you can imagine, gene regulation is a very important process. And being such an important process, you can imagine there are very many different ways to do gene regulation. So here are all the ones we're actually going to cover. So we're going to cover operons, histone modifications, transcription factors, alternative splicing, MI or microRNAs, protein activation, and protein degradation. And of course, there are many, many, many more ways in which you can regulate genes beyond this, but here we're covering the ones that your textbook mentions. So the first one we're going to cover is a lac operon. You actually find description of the lac operon in probably every major biology textbook. So it's actually one of the simplest examples of gene regulation, which is why it's such a common example. So functionally, the genes that make up the lac operon function to digest lactose. And that's going to be a very important information when we describe how the lac operon actually works. And so operons themselves are mostly found in bacteria and archaea. They're not very often found in eukaryotes. Also, at this point, I should probably stop and define what an operon is. So an operon is a collection of genes that serve a similar function and are regulated by some sort of operator. So that will make a little bit more sense once we go to the next slide, but for now, just know that definition. And in the bottom right-hand part of the slide, I have a picture of lactose. So lactose is a disaccharide made up of galactose and glucose. So essentially two sugar molecules linked together. Here specifically, galactose and glucose linked together. So here is a lac operon. As you can see, a very busy slide, so I'll walk you through this piece by piece. So the lac operon itself is made up of multiple parts. There's a promoter. As you can see, RNA polymerase is binding to the promoter, just as we saw in earlier chapters. There's the operator. We'll see what that does a little bit later. And then we actually have the genes that make up the lac operon. So we have lac Z, lac Y, and lac A. So again, an operon codes for genes that serve a similar purpose. So for example, something like beta-galactosidase, that actually breaks apart lactose. So it breaks it down into the monosaccharides, glucose, and galactose. Permease, or lac-Y, codes for a gene that actually allows for lactose to cross the plasma membrane. So it actually allows lactose to come into the cell. Transacetylase, we actually don't know what it does, at least the last time I checked. But here it is part of the lac operon, so it serves some function related to lactose utilization. And I said something just very important right there. So I said lactose utilization. So I should say the major function of the lac operon in general is to break down lactose. So that's going to play in a very important role, as we're going to see, in how the gene is regulated. So outside of the lac operon proper, so here lac operon extends from here to here. So outside of that, we have the lac I gene. So I equals inhibitor. So even though it has a lac prefix, it's still considered outside of the operon itself. So that gene gets transcribed in mRNA and is made into this protein right here. So this protein is often simply called the repressor. So it represses transcription of the lac operon. So normally when there's no lactose present in the cell, the repressor is going to be bound to the operator. So you can see it has this nice shape that actually fits directly into the operator. So you can think of the repressor almost being like a wall. So it acts as a wall to block transcription by RNA polymerase. So that's the case when there's no lactose in the cell. So it's bound to the operator and is blocking transcription of the lac operon. So it's blocking transcription of beta-galactosidase, paramase, and transacetylase. And you may be wondering yourself, why does the cell regulate the lac operon this way? So remember, the point of the lac operon is to utilize lactose. So if no lactose present, then why waste all this time transcribing and translating these proteins? So the lac operon should only be on when there's lactose present. So there's actually lactose that needs to be broken down. So that's the logic of the cell. It doesn't want to waste energy producing proteins it's not going to use. So it churns off the lac operon using this repressor. Okay, so now imagine there's lactose in the cell. So the cell needs some way to tell there's lactose present. So what happens is that lactose, here called allolactose, actually binds the repressor. So once it binds the repressor, it changes the shape of the repressor, and it can no longer bind the operator. So it no longer does this. And because of that, RNA polymerase can now transcribe the lac operon. So we have reduction of beta-galactosidase, paramase, and transacetylase, so that allows for utilization of lactose. So if you think about the purpose of the lac operon, I think that makes a lot of sense in how it's regulated. So when there's no lactose present, the repressor is bound, since you don't want transcription and translation of lactose utilization genes. When there is lactose present, though, you want the repressor unbound. So you want transcription and translation of those lac operon genes, since that's going to allow you to utilize lactose. And if you use that same logic, you can apply that to the tryptophan operon. So a tryptophan operon is actually called repressible. And I'll talk a little bit about the function of the tryp operon in a bit. But first, we'll go over the left-hand side of this figure first. So here again is a lac operon. So that's called an inducible operon. So it's induced by lactose. So lactose essentially turns it on. So when there's no lactose, the operator is bound. And when there's lactose present, there's an inactive repressor and it's not bound to the operator. The tryptophan operon, though, is different. So a bacterial cell is actually able to make its own tryptophan. So tryptophan is an amino acid. So when there's tryptophan present, the repressor is active and the tryptophan operon is not on. Okay, so let's think about the logic behind this. So tryptophan is an amino acid and the cell is able to make it. So if we have tryptophan present, then why would you want the cell to make more tryptophan? So that's exactly the logic here. So if there's tryptophan present, the repressor is bound to the operator. And that's because the cell doesn't want to make more tryptophan if it doesn't need to make it. However, if tryptophan is low, the repressor is unbound, and you have the tryptophan operon activated. So then you produce more tryptophan, and eventually you switch back to the top condition. So again, use your logic to figure out how these operons work, since I think that's probably the best and most intuitive way to learn them. Here's question 2 for Blackboard. This is a question that should be familiar to you from the lab we did on DNA technology. So DNA overall has what charge? So as you probably remember, DNA has an overall negative charge. And the reason why I bring this up is it will become important in the next few slides when we talk about histone modification. So as you probably remember, DNA is wrapped around histones. So histones are proteins, so we have DNA wrapped around them. Oftentimes, DNA wrapped around histones can be called beads on a string, which is what it actually looks like in an electron micrograph. So here you can actually see the histones and the DNA that's wrapped around them. Here is an artist's rendition of that. So we have DNA wrapped around histones. So here's a question for you. So if DNA has a negative charge, what charge do histones likely have overall? So if you think about this, opposites attract. So histones probably have an overall positive charge. And actually they do. So you can imagine if histones had a negative charge, DNA would not like wrapping around histones. But since they have a positive charge overall, DNA likes to wrap around histones. So now imagine what would be the effect in making histones more positively charged or more negatively charged. So what would happen to the DNA? So you might imagine that if we make histones more positively charged, DNA would be more attracted to it. So DNA would actually pack more tightly. But for a negative charge though, we're going to have DNA repelled from the histones. So DNA is going to become looser. And that's exactly what happens in these histone modifications. So the two major types of histone modifications are methyl modifications or acetyl modifications. So a methyl group has an overall positive charge, which means DNA becomes more tightly packed. Acetyl groups, though, have a negative charge, so DNA becomes less tightly packed. So again, remember opposites attract. So knowing this, how does this affect gene regulation? So remember, we can pack DNA more tightly or more loosely depending on how we want it. So in the most extreme form, we have chromosomes. So chromosomes are very, very tightly packed strands of DNA. On the other hand, we can have something very loose, like this DNA double helix right here, which is very unpacked. So really how histone modifications regulate DNA is in the packing of DNA. So you have something like this condensed chromosome right here. RNA polymerase can literally not reach the gene it wants to transcribe. So it's literally a space issue. So RNA polymerase just cannot fit anywhere and transcribe the gene. However, for something like the strand of DNA right here, which is loosely packed, RNA polymerase is easily able to access the gene it wants. So again, if we have more methyl groups and DNA is more tightly packed, RNA polymerase is going to have a hard time transcribing that gene. If they're more acetyl groups, though, it would be less tightly packed, and RNA polymerase could access the gene and transcribe it. So this is how histone modifications allows for gene regulation. And here's that just written out for you. So if it's too tightly packed, RNA polymerase cannot reach the promoter, so we have no transcription. If it's loosely packed, though, transcription can happen since RNA polymerase can reach the promoter. One important side note is that histones and DNA modifications can actually sometimes be inherited. and we call this epigenetic inheritance. So we won't discuss too much about that, but it's an important note to make since this is becoming a very important field. So an extreme example of histone modifications is X inactivation. So humans actually only need one X chromosome to survive. So for males, this is totally okay. We only have one chromosome by nature, but for females, this might be an issue. So why would having two X chromosomes be an issue for a female? So the answer here is dosage. So essentially, females have twice as many X chromosome genes as they need. And because of that, if we had two X chromosomes on, we'd have transcription and translation happening twice as much as normal. So this is an issue for females because of dosage, specifically dosage of the protein in mRNA. So histone modifications actually play a huge role in this X inactivation process. So this process, just as the name implies, turns off one of the X chromosomes. And histone modifications play a huge role in this process. So it actually turns out that every cell in the female body has one X chromosome turned off at random. And these inactivated X chromosomes are called Bar bodies. So we'll actually see a picture of this in the next slide. So in the early embryo, both X chromosomes are actually on. So here we can see we have two X chromosomes, completely normal. And after a few cell divisions, the X chromosome will actually become inactivated. So as you can see, in this cell, we have this chromosome inactivated. And in this cell, we have this chromosome inactivated. So this is X inactivation and creation of Bar bodies, so essentially inactive X chromosomes. So I told you this process was essentially random, so which X chromosome gets turned off is completely random. And this is actually how calico cats are created. So going back to the left-hand side of this figure, we have one chromosome with an allele for orange fur. The other X chromosome, though, has an allele for black fur. So again, which X chromosome gets turned off is completely random. So some will have the black fur chromosome turned off, which would mean we have orange fur. And some will have the orange X chromosome turned off, which means we have black fur. So it's a completely random process, and that's how we get a calico cat like this one right here. So it's X chromosome turned off and on randomly in early development, and this led to the pattern of their coat. So here's the third question for Blackboard. So can a calico cat ever be male? Well, the answer here is no. So remember, males are XY, so they don't have that dosage issue. So we only have one X chromosome, which is enough for us to survive. So it's only females with two X chromosomes that undergo X inactivation. So all calico cats are going to be females. So the next form of regulation we're going to talk about are transcription factors. So transcription factors are proteins that help recruit RNA polymerase to transcribe a gene. It's important to note, though, that transcription factors can also silence genes, or essentially turn them off. So transcription factors can act as activators or silencers. Oftentimes, students will forget the silencing part and think that transcription factors always activate genes. So it's not true, they can also act as silencers. So in the next slide, we'll actually see how transcription factors work. So here's one model for how transcription factors might work. So transcription factors normally bind to pieces of DNA called enhancers. So enhancers can really be anywhere in the chromosome, and in some extreme cases on different chromosomes. But essentially they are the regions where transcription factors are going to bind. Here we have our transcription factors, and as you can see, there are quite a bit of proteins involved here. But essentially the main gist of this figure is that they all combine and bend the DNA to recruit RNA polymerase. So this is actually one of the leading hypotheses about how transcription factors work. So through this bending of DNA that links together enhancers and promoters, that actually allows it to recruit RNA polymerase. The next form of regulation is alternative splicing. So remember in RNA processing, which we talked about in the last section, introns are removed and exons are spliced together. So in alternative splicing, we have various exons excluded or included during the process of intron removal. So we'll talk more about this in the next slide, but know that the function of alternative splicing is to allow us to produce a variety of different proteins from the same messenger RNA. So here we have DNA, we have 1, 2, 3, 4, 5 exons and some introns. That, as you remember, go through the process of transcription. So we have RNA produced, and then RNA has 1, 2, 3, 4, 5 exons in it. So as normal, the introns will be removed, but here we have alternative splicing. So depending on how the RNA is spliced, we can either get protein A or protein B. So protein A contains exons 1, 2, 3, and 5. Protein B contains exons 1, 2, 4, and 5. So as you can see, alternative splicing allows us to produce different proteins from the same messenger RNA. And there are many more combinations, just the two shown here. So you could have exon 1 by itself, exon 2 by itself, 1, 2, and 3, 4, 3, and 1, so on and so on. So you can see there are quite a bit of combinations here, and this allows for a lot of flexibility for the cell in what proteins it produces. So next up we have microRNAs, sometimes abbreviated miRNAs. So these are short 22 to 23 nucleotide pieces of RNA. So notice I used RNA, not messenger RNA. So microRNAs are not translated into proteins. So they just stay RNAs. So these microRNAs are actually complementary to certain regions of messenger RNA. So again, messenger RNAs are the RNAs that code for proteins. So these microRNAs are actually complementary to certain regions of mRNA, and because of this, they can bind to messenger RNAs and either cause degradation or block translation. And the reason for this is because the cell does not like double-stranded RNA. So if a cell sees double-stranded RNA, it thinks that's bad and it will try to destroy it. So when we have a microRNA bind to a messenger RNA, the cell recognizes this and starts to destroy the messenger RNA. So here's that, just shown in figure form. So in addition to the microRNA, we also have a protein added to the microRNA that facilitates this process. So it's this microRNA protein complex that binds to a target mRNA. So again, remember that microRNA has some sequence that that's complementary to the messenger RNA. So it does this, and it either leads to degradation of the mRNA, or it blocks translation. So essentially, either way, we have no protein produced from this messenger RNA. Next, we have protein activation. So this is fairly intuitive. So some proteins are not active until properly processed. A very common example of this is insulin. So for insulin to be active, it actually needs to be cleaved. Essentially a fancy way of saying cut. In addition, we also need disulfide bonds produced. So both these processes are actually shown in the picture below. So first, we have normal translation of insulin. It looks like something like this. So sulfur hydrogen bonds all along the protein. So these bonds are actually linked together to create disulfide bonds. So as you can see, this greatly changes the shape of insulin. The next step is we actually have insulin cut. So this light purple part gets cut out, and you have something that looks like this right here, which is the active form of insulin. So again, this is protein activation. So without doing any of those steps, the protein would not be activated and not usable by the cell. So the cell can actually change how this happens, or if it happens at all, and it can regulate proteins by allowing them to not become active. On the flip side of this, we have protein degradation, also very intuitive. So any sort of protein that's no longer wanted is destroyed by the cell. So as a reminder or a review from earlier in the course, what organelle does a protein likely become activated in? So remember, the Golgi apparatus does protein modifications. So for something like insulin, that's likely modified in the Golgi apparatus. So I like this figure from your textbook. It goes over essentially everything we've talked about so far. So the first thing we have is DNA unpacking. And this is due to histone modifications. So methylation, which has a positive charge and packs DNA tighter, or acetylation, which has a negative charge and unpacks DNA. Depending on how tightly DNA is packed, that either allows or denies access of RNA polymerase to the promoter. So next, we're going to look after transcription. And after transcription, we can have splicing. So we have alternative splicing, so different exons are spliced together. We also have addition of the cap and tail. So this is something we didn't talk about, but the cap and tail can also be used to regulate RNA. So without the cap and tail, the RNA is actually very unstable, so it needs that cap and tail, or else it's going to quickly disappear. The cap and tail are also important for flow through the nucleus, so without the cap and tail, the messenger RNA would actually have issues leaving. Once the messenger RNA has left the nucleus, we can now have breakdown of it, likely through the process of microRNAs. So those are short, complementary pieces of RNA that bind the messenger RNA and cause degradation or inhibit translation. Now we're going to look at steps that take place after translation. So one way in which proteins can be regulated is through protein activation. So we talk about insulin, and also protein degradation or breakdown of the protein. So again, this is a very nice figure. So you can see they've chosen to use valves right here. So you can think of the cell churning these valves, turning them on and off to help regulate what genes are turned on and what genes are turned off. So this is exactly what's happening in the skin cells and intestinal cells that you have in your body. They're turning genes on and off, and that dictates their function, morphology, and everything else about them.